So we have already seen in my previous video that the process of editing which is occurring via deamination. So let me revise once again. What is RNA editing? How it is done? Insert, change, delete. What? A particular base. The process, what are the mechanisms by which editing happens? That is one, deamination. So deamination is of two types. One is cytosine deamination and the other one is adenosine deamination. We have already seen about cytosine deamination, where CAA, this particular codon, where a particular nucleotide is being chosen and this is deaminated, that is C changes into U and the rest of the two nucleotide remains the same, where CAA codes for glutamine and UAA is a stop codon. So where does this takes place? It takes place, this particular editing takes place in a particular tissue, that is intestinal tissue. So that is the reason we say this site specific deamination. But in liver shell we are not being able to witness this type of editing. So in liver shell we get a full length protein, whereas in intestine, where this kind of editing happens, we get a truncated or a smaller version of this protein. This example is seen, these two forms, which kind of protein these are? These two forms of apolipoprotein B are involved in lipid metabolism. And the information which was required, that this kind of deamination is done by a particular enzyme, we should not forget this also, cited in deaminase. Okay, now what is the other type of deamination performed? That is adenosine deamination, which is done, we can absolutely see adenosine, that is A is being A or adenosine, this particular nucleotide is changed into inosine. Now this inosine base pairs with the cytosine. How this A changes into inosine? With the help of a particular enzyme known as ADAR, adenosine deaminase, acting on RNA. Now this kind of deamination, this type of editing is essential in our brain, where the ion channel is being expressed. If this type of editing does not happen, it can impair or severely hamper our brain in functionality, and this type of editing is required because it alters the Ca2 plus permeability of the channel. Thank you, and don't forget to like and subscribe.